Okay, so now in chapter 6, we're talking about tertiary structure, and going from secondary structure to tertiary structure is something we call protein folding. So just to review where we are, there's primary protein structure, which is just the sequence of amino acids, and it's held together by peptide bonds. And again, this is the only covalent bond in this structure. Then there's secondary structure. Secondary structure is usually an alpha helix or some sort of beta pleated sheet. There's more examples, 310 helix. But these are the most common types of secondary structure. These are held together by hydrogen bonding within the backbone. So now tertiary structure. This is a three-dimensional structure. This is usually at the level that we call protein folding. And there's two types of tertiary structure. There can be fibrous, which we already talked about. And then there's globular, which we'll talk a lot more about later. And these are held together by hydrogen bonds in the R groups. So all of these are hydrogen bonds. Actually, let's not use hydrogen bonds. Let's say interactions between the R groups. And when we're talking about the types of interactions, the most common interaction between the R groups that we're going to have are hydrogen bonding. But also we're going to have what are called van der Waals interactions. And we also are going to have one other kind of covalent interaction, which is called a disulfide bond. These are only between two cysteine residues. We'll see examples of that where two cysteine residues can form a disulfide bond between each other. You might also hear van der Waals interactions, sometimes called ionic bridge interactions, and a lot of this is also going to be caused by hydrophobic hydrophilic interactions. So for a protein to be functional, for it to be able to do its job, it needs to be in the correct three-dimensional shape. So a protein isn't functional until it gets to this level. Even if the sequence is correct and the secondary structure is correct, it can't do the job it was designed to do until it is folded correctly. So all of this, we're going to start talking about protein folding. And protein folding is really just the process of going from an unfolded protein to a complicated three-dimensional shape that is going to be in the correct conformation for this protein to do the job that it is supposed to do. So let's talk about some of the jobs that proteins have. We already talked about fibrous proteins. We said that their job was mostly in structure. That was the collagen example that we looked at. When we're looking at globular proteins, which we haven't talked about yet, we'll talk about next. They carry out most of the chemical work in the cell. They're involved in synthesis. Globular proteins include enzymes. Enzymes are catalyzing reactions. An enzyme is just a protein that increases the rate of a reaction. It has catalytic activity. We're going to talk a lot about these later. All enzymes are proteins except for one, and that is the ribosome. The ribosome acts like an enzyme during protein synthesis. All other enzymes are proteins, and all chemical reactions in our bodies are catalyzed by enzymes. Remember, enzymes don't change the free energy of a reaction, they just increase the rate of that reaction. None of the chemical reactions in our cells would happen quick enough to support life, and enzymes are responsible for increasing the rate of chemical reactions. We're going to talk a lot about enzymes later. Globular proteins are also involved in transport and metabolism. We're going to talk a lot about globular proteins. Globular proteins are antibodies. This is probably the class of molecules that we'll spend the most time talking about this quarter. So what's important is that we get from this sequence of proteins to the correct folded structure so that these proteins can do all of these jobs that are very important. So there are important properties that determine the tertiary structure, this folded structure, and that is these hydrogen bonding van der Waals. These are all between the R groups. So how are these R groups of this long polypeptide going to interact with each other to form the lowest energy protein? And that's the shape of the protein. It's the protein shape that is going to have the overall lowest energy. That means all of these hydrophobic R groups, all of these hydrophobic amino acid R groups are going to be tucked deep inside the protein, not exposed to the aqueous solution outside it, because most of these proteins are existing in an aqueous solution somewhere. All of the hydrophilic amino acids are going to be on the outside of the protein, exposed to water. Hydrogen bonding is going to happen between the amino acid R groups. We're not talking about hydrogen bonding between the backbone anymore. We're talking about hydrogen bonding between the R groups. So hydrogen bonds will form. We'll have van der Waals interactions between positively and negatively charged amino acids. We'll have disulfide bonds between cysteines. And that is going to form the protein with the lowest free energy. So we are pretty good at predicting what a folded protein structure is going to look like from the sequence of the protein. And this is called the Leventhal paradox. And when you look in your textbook and on the PowerPoint slides, it's going to show you what's called an energy landscape model. And it kind of looks like a funnel. And down at the bottom of that funnel would represent the lowest free energy. And what actually happens is, instead of it looking like a real funnel, on the way down to the bottom, there's all these ridges. It kind of looks like a mountain range in there. And what's happening is there's actually some good animations of this. If you just Google Leventhal's paradox, protein folding, energy diagram, something like that, it'll show how a protein will kind of reconfigure itself and fold and unfold and refold until it's reached the conformation that has the overall lowest free energy. And that lowest free energy state is going to be the three-dimensional structure of that protein. So something that I want to make sure we understand is that there is a difference between proteolysis and denaturation. So let's see. So what proteolysis is, again, the name kind of defines what's happening. Proteo means protein. Lysis means breaking. This proteolysis is cleaving the peptide bond. So this would be breaking primary structure. Denaturation is really unfolding the protein. So this would be breaking or disrupting secondary structure through quaternary structure, if there was quaternary structure. We'll talk about quaternary structure later. This is going to be disrupting all non-covalent interactions and those disulfide bonds to unfold the protein. This is different than breaking apart the protein. Both of these will result in a protein that's not functional. An unfolded protein is not functional. But if you're cleaving the peptide bond, that's physically breaking the protein apart. So when we're talking about denaturing a protein, that's different than proteolysis, which is cleaving the peptide bond. Once you denature a protein and you unfold it, you can then do what's called renature. I'll say renaturation, which would be refolding the protein. And there's all kinds of experiments that were done a long time ago for us to understand how a protein can be denatured and then renatured, refolding. It turns out all of the information that a protein needs to fold into its functional form is in this primary sequence. So if you denature a protein in the correct way, you also could then renature a protein and then get what's called the nascent or the functional form of that protein. So there's all kinds of examples of experiments that were done for us to understand that, and I'm just going to have us talk about one together. And the one that I'm going to have us talk about, let me just get to where it is on the PowerPoint slide so you guys can refer back to it. It is slide It's the slide where you're denaturing with an organic solvent and then reducing the disulfide bonds. Hang on just a second. Lost the slide. Okay, it is slide number 37. And it's an experiment with a protein called ribonuclease A. So the protein isn't important, but that's what protein this is. And it shows that when you have a native molecule, native meaning correctly folded, and there's two things that's happening. You're reducing it with BME, beta-mercaptoethanol, and then you're denaturing it with urea. So reducing it with BME, so maybe I will draw it over here. So we have this protein that's folded correctly. I'll say this is the native molecule. And then a few things were happening. One, they were reducing it with BME, and that is going to break the disulfide bonds. And then in this order, the order is important, they were denaturing it with urea. Urea will unfold the protein. And then you get kind of this unfolded protein. They learned that you could get back to this native state if you do it in this order, remove the urea, which is going to cause the protein to fold again, and then oxidize, which will cause the disulfide bonds to reform, you would get this native structure again. They found that if you did the opposite, if you oxidize first and then you remove urea, you do not have a functional protein. You have something that's kind of folded, but you have random disulfides bonds in here because you oxidized first, and this is not the functional protein. So we know that you can denature a protein and then renature it again. So let's talk about some of the denaturing products, ways to denature a protein. Denaturing conditions include increased temperature, change in pH, very acidic or very basic, or organic solvents like urea, for example, in the example that we just did. So that's how you denature a protein. A way to proteolysis, when you're actually cleaving those peptide bonds, this is usually catalyzed by enzymes called proteases. Anytime you see the suffix ase, that indicates that there is an enzyme there. So there's a difference between denaturing a protein and proteolysis, which is cutting that backbone by breaking that peptide bond. So there's lots of examples in the textbook and in the PowerPoint lecture. We are going to spend a lot of time talking about enzymes and proteolysis later, so we don't really need to spend a lot of time on that here in this chapter. One other thing that I'd like you to pay attention to in this chapter are chaperones. I'm not going to draw out the chaperones because it's kind of complicated structures, and there's some nice pictures in the textbook and in the PowerPoint slides. So pay attention to chaperones. And the images for this uses the GROW-EL, GROW-ES chaperone system. This is slide number 58. And what these chaperones are, it's two complexes that essentially makes a barrel-like. You can almost think of it like the inside of a washing machine. And what happens is sometimes these proteins don't fold by themselves. They need some help. And that's exactly what a chaperone is. So the chaperone provides an environment that will allow this protein to fold correctly. So this protein would enter this chaperone, and it's kind of like a washing machine that shakes it all up inside, and then it releases the properly folded protein structure. So there's pictures of that in the textbook and in on the PowerPoint slide number 58. One thing that is really important about protein folding is that when proteins do not fold correctly, there are processes in our bodies that should recognize those misfolded proteins and destroy them. But there is a whole class of diseases called amyloid-related diseases that result in a misfolded protein that does not get destroyed. And these are diseases like Alzheimer's, Parkinson's, ALS, Huntington's disease, type 2 diabetes. These are some of the worst diseases for us to deal with because there's no way for us to combat these diseases because it's not like an exterior pathogen that we're trying to fight. It's our own healthy, correctly sequenced protein that is misfolding. So these are called amyloid-related diseases, and they are on slide number 59. We'll kind of refer back to this later when we start talking about disease processes and stuff like that. There's also a lot of biochemistry in practice at the end of chapter 6, talking about molecular cloning and all kinds of different techniques that we can use with proteins. Definitely look through these, read the parts about this in your textbook about protein purification. We will come back to these in different portions of the course, but this is not something that I'm going to quiz you on this week or include on the first exam.